Good morning, students. Today, online class on classification of virus. So, virus are classified on the basis of habitat. That means it is present on the coast plan, which is they are based on the structure, chemical composition, similarities of genetic makeup. International Committee on Taxonomy of Viruses, which includes three orders, 63 families, the family ends in Viridae, 263 genera. So, a classification based on the structure, virus structure, chemical composition. What are the chemical composition like? There will be nucleic acid, protein, and what are the capsid, capsomere present, and then there is the genetic makeup. What are the gene concept present in this particular virus? Then, so based on this, the classification or viruses are classified. They according to three orders are there. The first orders are they are called as the first order cordovirus, then mononegnovirals, then another one is nudovirals. So this another one slide shows the types of classification. There are three types based on the classification. First one is Baltimore classification. David Baltimore, in the 1971, he classified the virus based on the mechanism of RNA production, the nature of genome, and the nature of genome. So this was mostly based on the genome. Then second one is classical system of classification. Third one is genetic classification. So first one is detailed study about the Baltimore classification. So the Baltimore classification gave the 1971 the viruses are classified on the basis of mechanism RNA production, the nature of genome. The genome is either single stranded or double stranded. So according to Baltimore, there are seven groups are there. The second groups are classified based on the genome replication. So the Baltimore classified the RNA as well as the first one is dsDNA viruses. The dsDNA viruses are double-stranded DNA viruses. Example for these are adenovirus, herpes virus, and pox virus. Second one is then the second one is single-stranded DNA virus. The single-stranded DNA virus, they are single-strand DNA viruses are present in the parvoviruses. Third one is double-stranded RNA viruses, example reoviruses. Then fourth one is plus or RNA viruses plus strand the sense via RNA. Example, the Toga viruses. Then fifth one is minus double strand RNA viruses. So double S single stranded RNA viruses. The viruses may be RNA either minus strand or antisense RNA. Example for rhabdoviruses. Then sixth one is double stranded then single-stranded RNA viruses. The strand may be plus strand. They are sense with the RNA. Or another one is intermediate life cycle. That is retroviruses. Then seventh one is double-stranded DNA viruses. Example for Keptida virus. So the Keptidana virus is the seventh DNA viruses. So Baltimore classified the virus into seven types. So, what are the importance of the molecular weight examples? Then, what are the family? Then, for the seven types of virus. First one is the viruses detail. The another one is animal viruses. The animal viruses on the animal coast plant. They cause disease like rabies, polio, mumps, chickenpox, smallpox, and influenza. The plant viruses which show Their leaf characteristics when attached with the plants. So they present only the plants. Tobacco mosaic virus DME and banana streak virus, carrot thin leaf virus. So all these are including the plant viruses. Then bacterial viruses are bacteriophages, bacteria infecting viruses that the viruses infect bacteria is called bacteriophages. They are T1, T2, T3 and T4. So already you studied the structure of T4 bacteriophages. Then the structure shows the T4 bacteriophages a tadpole shaped, presence of head and tail, collar and base mass with the tail fibers, and then they will be having a central DNA nucleic acid surrounded by a protein coat. Another is tobacco mosaic virus. The tobacco mosaic is having a rod-shaped structure, it is having a helical like structure, but it is having a RNA, but it will be surrounded by a capsome. Here is a capsid is a unit of subunit of protein. Then another one is classification based on genetic basis. According to genetic basis, the viruses are classified into DNA viruses and RNA viruses. Genes and the genes எப்படி இருக்குன்னா மேபி லீனியர் ஆர் சர்க்குலர். சில வைரஸில் வந்து நீளமாக இருக்கும், சிலதில் வந்து வட்ட வடிவத்தில் காணப்படும். The smallest have only 4 genes and largest have several hundred. So இந்த சின்ன இதில் வந்து சிறிய இதில் வந்து எவ்வளோ இருக்கும், 4 ஜீன்ஸ் இருக்கும். அதில் பெரிய இதில் வந்து 100க்கும் மேற்பட்ட ஜீன்கள் காணப்படும். டிஎன்ஏ வைரஸஸ், த டிஎன்ஏ வைரஸஸ் ஆர் த வைரஸ் விச் கன்சிஸ்ட் ஆஃப் டிஎன்ஏ. ஸோ டிஎன்ஏ வைரஸ்னால் அதில் என்ன இருக்கும், டிஎன்ஏ இருக்கும். தே கம்ப்ளீட் தேர் ஆக்டிவிட்டீஸ் டிரான்ஸ்கிரிப்ஷன் ஆஃப் மோஸ்ட் ஆஃப் த அட்டாக் ஆன் சிம்லர் ஜீனோம். ஸோ சிம்லர் ஜீனோமில் தான் என்ன பண்ணும், இது அட்டாக் பண்ணும். ஆர்என்ஏ வைரஸஸ் சொல்ல வந்து the RNA viruses are the viruses which consist of RNA genome. They complete their activities the reverse transcription. So reverse transcription la dhan idu life cycle complete agum. Then another one is the important for DNA virus groups. So what are the DNA virus groups are present in these DNA viruses? Then classifications based on the structural basis. On the basis of structure, the morphology of viral structures, viruses are having a envelope as well as non-envelope viruses. So some of them are enveloped, covering, another one is not covering. So however, they are arranged in subclasses of DNA and RNA viruses. Then in genetic engineering, the viruses are used as genetic vectors which carry good genes to cells. So genetic engineering, it is a carrying of vectors which is very important for introduce new genes into the cells. Viral envelopes stimulate the immune system to produce antibodies that can protect the host cells against future infections. So the virus வந்து என்ன பண்ணும்னா நோய் தடுப்பாற்றலை அதிகப்படுத்தினால உள்ள ஆன்டிபாடிஸ் அதிகமாக ப்ரொடியூஸ் பண்ணி கோஸ்ட் செல்லில் வந்து ஃபர்தராக இன்ஃபெக்ஷன் ஆகாமல் தடுக்கிறது. Viral genome contains certain enzymes for specific operation within the host cells. சில வைரஸ் ஜீனில் வந்து குறிப்பிட்ட என்சைம்கள் அந்த பர்டிகுலர் ஸ்பீஸ் கோஸ்ட் மட்டும்தான் ஆக்டிவ் ஆகிடும். ஸோ ஆன்டிவைரல் ட்ரக்ஸ் பிளாக் வைரஸ் ரெப்ளிகேஷன் பை டார்கெட்டிங் ஒன் த ஸ்டெப்ஸ் தி வைரல் லைஃப் சைக்கிள். தென் தி இன்டர்ஃபெரான் இஸ் த protein potential for treating and preventing viral infection. Then recently developed drugs to combat some viruses, mostly interfering with nucleic acid synthesis. Then the ACT interferes with the reverse transcription of HIV. Then acyclovir inhibits herpes virus DNA synthesis. Okay, thank you. That's all. These are the virus classification. So according to the Foltimer classification is very important. There are 7 classes, one by one. Double stranded DNA, virus with single stranded DNA, virus with double stranded RNA, virus with plus or sense single stranded RNA, virus with minus antisense single stranded RNA, virus with plus sense RNA or RT that replicate DNA intermediate in the life cycle, virus with double stranded DNA RT that replicate with the RNA intermediate in the life cycle. So what are the types of virus based on the Foltimer classification? Okay, thank you.